All right, hello everyone. This is our topic 4.5 in our cell communication unit. This topic, 4.5, is primarily talking about the cell cycle. And so how does a cell basically replace damaged cells? How does cells divide to grow and develop? And so we'll look at all of those like step-by-step processes in prokaryotes, and then we'll take a deeper dive into eukaryotes to see how that might be different. So to start, for prokaryotes, we are just thinking about like the most simple organisms. They don't have a lot to split up. It's really just their genetic material. So these are organisms without a nucleus. We know that. Their genetic material is arranged in a single circular chromosome of DNA. The DNA is duplicated before division. And then prokaryotic cells, in addition to their circular chromosomes, also have what we call a plasmid. Plasmid is a small circular DNA molecule in the cytoplasm. Now, this plasmid kind of behaves or replicates independently of the other chromosomes or the circular chromosomes. You should know, too, that prokaryotes have circular chromosomes. So if you see that, that's kind of like a sign that we're talking about prokaryotes as opposed to eukaryotes. And the plasmid contains a small number of genes. This will also be talked about in Unit 6. These genes can help prokaryotes fight off like antibiotic resistance genes. So they help fight off other bacteria. And so plasmids are most commonly found in bacteria, but can also be present in a few eukaryotic organisms like yeast. And for prokaryotes to divide, they basically elongate until they double their size. The cell pinches in and separates into two identical cells in a process called binary fission. So we can see that over here. Basically, all of that genetic material needs to divide and duplicate itself. So that circular chromosome we see here kind of elongates and separates until eventually the cell is able to kind of cut its cytoplasm in two and make two unique cells. We can also see the plasmids are dividing over here as well. So it doesn't get a lot more complicated for that for prokaryotes. Eukaryotes is where it kind of is more complex. And so eukaryotic cells go through this cell cycle, which is the life cycle of a cell, a series of steps where the cell prepares for division and proceeds to divide. We need to know this graph or this model here of the cell cycle. I would remember this. Remember the steps in order, which ones are in interphase, which ones are in cell division or the M phase, and what they do, and also what they look like on the cells. So the entirety of the cell cycle is made up of two steps. We say interphase, and then cell division. And so we start with interphase. Interphase is the period where the cell is preparing for division. It's growing in size. It has to duplicate its DNA. It has to duplicate its organelles. It has to be a large enough size to be able to divide. And so it's kind of like preparing for all of these steps for when it actually goes through the cell division phases. So interphase is kind of like the cell in a normal state with a nucleus. We also talk about, if you look at the graph, there's this section over here where it kind of looks like it goes off to like the side. This is called G0. And so G0 is where cells are not actively dividing or not actively preparing for division. So many cells can just stay in G0 for a long time and don't need to divide in the organism. But if a cell does need to divide to replace damaged cells or just to develop or grow an organism, it's going to involve three stages. So the first step in interphase is G1, where the cell is just growing. The second step is the S phase. S stands for synthesis. This is where all of the DNA needs to copy itself. All right, so if we look over here, we can kind of see two unduplicated chromosomes. And then after the S phase, we have two duplicated chromosomes. So if we were kind of counting the amount of chromosomes, we'd say there's two chromosomes here, there's two unduplicated chromosomes here, and then there's still two chromosomes here. So it's still 2 to 2, and that is going to be important when we start counting our chromosomal number. But we just say we go from two unduplicated to 2 duplicated. So you go from 46, for humans, you go from 46 unduplicated chromosomes to 46 duplicated chromosomes. And this is still in the form of chromatin, which we'll talk about in a second. And then our last step in interphase is G2. The cell is still growing. The cytoplasmic components are doubled. So like organelles are duplicated during the G2 phase and prepare for division. So you can kind of see a cell spends a majority of its time in interphase, and once it gets to the actual cell division part, that happens quite quickly, but it's kind of also the more where more of the content is and where more of the vocab is that we need to know about. But in interphase, inside the nucleus, the DNA and genetic material is in the form of what we call chromatin. So chromatin is uncondensed genetic material made up of nucleosomes. And a nucleosome is basically, chromatin is just two things. So it's really just one thing if you want to just say it's a bunch of nucleosomes. A nucleosome is just made up of two things, which is DNA wrapped around a histone protein. And so we can kind of see that here. So the histones are these kind of taking up space. This whole thing is one nucleosome. And the histone's function is primarily for the DNA to take up less space, right? You know, when you wrap up a wire or wrap up your charger, you wrap it around a thing a bunch of time just to organize the DNA and to basically be able to activate the DNA or inactivate the DNA. When it's all spooled up like this, we would say this is like inactivated, like an inactivated gene. And then as it starts to unwind, kind of like that section, that's like DNA that's available to be transcribed, that would be activated. And so the chromatin is eventually going to start to condense into what we know as the chromosomes. Right? And chromosomes are those X-shaped genetic material in the cell or in the nucleus. We can kind of see that as well here. The DNA is wrapped around the histones. We have the nucleosomes, and then all of that condensed makes up a section of the chromosome once it condenses. Okay, so let's talk a little bit about the cell division steps. So this is also known as the M phase. There are two main phases in the M phase. So there's mitosis, which you might be familiar with way back when you first took biology. And the second one is cytokinesis. So hopefully some of this is kind of a review. We really don't we go in the same amount of depth in bio as we do in AP Bio, so it will be kind of a lot of the same stuff. And so mitosis is all about dividing the cells and for cell division. So specifically for mitosis, we're dividing the nucleus. We take one nucleus, we break it up, we send the chromosomes to opposite sides of the cell, and then we form two new nuclei at the ends of the cell by the end of mitosis. This is going to occur in 4 steps. We call them PMAT. That's the order that they take place in: prophase, metaphase, anaphase, telophase. There is also it's kind of like a half step in here that you'll sometimes see. It's called prometaphase. So we can take a look at that at some point, but that kind of exists as another step. But we have interphase here, kind of just looks like a normal non-dividing cell. It's just a cell in its normal state. As prophase happens, we can start to see the chromosomes form. Metaphase, they move along the middle. Anaphase, we're separating those chromosomes. Telophase, we're forming two new nuclei around them. And so we're gonna kind of walk through those steps just to kind of see it a little bit more in detail. So our first step in mitosis, so first step in cell division as well, is prophase. Prophase, a lot of things are kind of taking place, so there's a lot of vocab in this topic. First is that the nuclear envelope breaks down, so that's going to be, You kind of see the dotted lines there to show that it's breaking down. The chromatin, as we mentioned. So I always think of chromatin. This is kind of chromatin right here, actually. Let's do something like that. So it's just like a bowl of spaghetti. It's just kind of all spooled up together, no specific shape. We wouldn't be able to see chromatin underneath a microscope. It's not condensed enough. But chromatin is going to start condensing into chromosomes. And one chromosome is made up of two sister chromatids. And so you can see that here. So this is one sister chromatid. This is another, blue is a bad choice. This is the other sister chromatid, and those make up one duplicated chromosome. Sister chromatids will be genetically identical unless there are mutations, but whatever gene is on this side, we'll talk about genetics in the next unit, but maybe that's like big A. This sister chromatid will also have big A. And in the middle of the sister chromatids, the things that connects them is our centromere. So centromere is the center of the chromosome connecting our sister chromatids. Okay, so we also have sister chromatids, we have chromatin, very similar sounding vocab that we have to distinguish. And then we also have the centrosomes. So they're centromeres, they're centrioles, they're centrosomes. A lot of stuff going on. But the centrosomes is comprised of two centrioles, and those are going to move to opposite poles of the cell. And the centrosome is where the spindle fibers, microtubules, form from centrioles and anchor them to those opposite sides. So let's take a look at some of that. So we have the two centrioles right there. This whole section is called the centrosome, the area where the centrioles and the spindle fibers take place. Spindle fibers coming out of those centrioles. The centromere is in the middle of the chromosomes. Our chromosomes are made up of two sister chromatids. Our nuclear envelope is breaking down. So a lot of stuff, right? A lot of vocab. So all of this is taking place during prophase, right? We're kind of like getting our early division started. Next is that phase I mentioned, prometaphase. And so spindle fibers are made out of microtubules, and they're basically going to move towards the chromosomes from the centrosomes, as you can see over here. And then this is also the formation of the kinetic core. So the kinetic core is a protein in the middle of the chromosomes on the centromere, and it's going to be where the microtubules will be able to attach to the chromosomes. So it kind of provides like a hook. So like this section in red is the centromere, and then the pink structures you see here, those are the kinetic cores coming out of the centromere, so the spindle fibers can attach to them. And so we're just doing in this phase, we're just latching on to the chromosomes so that we can pull them apart later. Our next phase is metaphase. This one's very quick. The chromosomes line up along the metaphase plate. You also might see this as equatorial plate, like equator. Same thing though. It's just the middle. So metaphase, middle, easy to remember. So if you see a bunch of chromosomes lined up, that's metaphase. The spindle fibers attached to the kinetic core. And now we get to our third phase, anaphase. And this is where we actually start to separate things. So, shortest stage of mitosis, separating sister chromatids and pulled to opposite sides, two centrosomes by microtubules. So these microtubules, the spindle fibers, are pulling the chromosomes to opposite sides. So, big idea. We are going from duplicated chromosomes to two duplicated chromosomes to two unduplicated chromosomes as soon as the sister chromatids separate. So it's still the same number. Mitosis keeps the chromosomal number the same as cells divide. So if we start with 46 chromosomes in a cell, the cell divides, we should still expect 46 chromosomes in both of them. We just say during mitosis, we go from 46 duplicated chromosomes to 46 unduplicated chromosomes in each one. So we can kind of label that duplicated. And then these two are both unduplicated. All right, we replicated our DNA during the S phase, which is before mitosis, but we still want to keep track of like, how do we have enough DNA for all of these cells? It's because we already doubled it in the S phase. So 46 to 46. You have to make sure you know that. It can get kind of confusing once we introduce meiosis at a later time. But our chromosomes have moved to opposite sides of the cell. We now get to telophase, which is basically going to undo the steps of prophase because our chromosomes have moved to opposite sides. We now need to form two daughter nuclei. So that's a key thing that happened to distinguish it from telophase. In the almost divided cell, chromosomes become less condensed, eventually forming chromatin, right? So hopefully that's sounding a little more familiar. On opposite sides in two new nuclei, the spindle fibers, or the microtubules, are broken down, and mitosis is done. This is not the end of cell division, just the end of mitosis. We still technically have one cell, right? The cytoplasm has not yet completely separated. And so that takes us to cytokinesis, division of cytoplasm to complete cell division. This is ensuring equal distribution of cytoplasm to both daughter cells, so basically that they're the same size. And so that happens, and then cells can enter the G0 phase where cell division no longer occurs. A cell can re-enter division with appropriate signals. So whenever there's a need to make more cells, cells kind of know when to do that. Cells, or different types of cells, divide differently. In animal cells, cytokinesis involves the formation of what we call a cleavage furrow, which pinches the cell in two. So we can see that here it kind of is just that indent between the one cell that's going to make it two. And then in plant cells, the cytokinesis involves the formation of a cell plate. So they have to work around the cell wall. And so once the chromosomes move to opposite sides, we have the cell plate form, like dotted lines, and it gets a little sturdier and eventually forms two new cell walls for those two new daughter cells that have formed. And that's our last step of cell division and the last step of the cell cycle. So we can kind of follow mitosis and cytokinesis here. So you can kind of see all of the steps. This is a good model or picture to study as well. Okay, so if we look at the check-in questions here from a video, we can kind of see how much the average amount of time that a particular eukaryotic cell spends in each phase of the cell cycle. Based on figure 1, what % of the time required to complete a full cycle do the cells typically spend in interphase? So the key understanding for this question is to know that interphase is G1, S, and G2. If we add all of those up, we should get 19. Right? And so 19 hours were spent in interphase, and then that was the total amount of time is just added the cell division, which was 1 hour. So we have 20 total hours spent in the cell cycle, 19 of which was in interphase. And so we just do 19 divided by 20, and that would give us 95%. And so a cell spends most of its time in interphase. If we look at the second check-in question, figure 1 shows the number of chromosomes observed in an actively dividing human cell at each stage of cell division. Which of the following best describes a correct interpretation of the changes in chromosome number depicted in figure 1? So we have prophase. It looks like we have about 46 chromosomes. This looks like a human. Metaphase, we have the same amount. And then anaphase is where looks like it jumps up. The number of chromosomes per cell, technically, we would say unduplicated chromosomes at anaphase, right? We go from duplicated to unduplicated, duplicated to unduplicated. And so I'll give you a second to look at the options. And what we should see is that C is going to be the correct answer. So chromosomes enter metaphase containing 2 chromatids attached by a centromere. During anaphase, the chromatids are separated, each becoming a chromosome. So that's the phase. Anaphase is when they specifically become their own chromosomes. Cytokinesis distributes the chromosomes into 2 separate cells. Okay, so there's some questions to go with this, a lot of vocab, a lot of memorization, and then once you do that, I think the analysis part of this topic is fairly straightforward. That was topic 4.5, and I hope that was helpful. Thanks.